Standard chemotherapy relies on saturating the entire circulatory system. The chemicals flood the bloodstream, forcing their way into almost every tissue in the body. The math behind this method is grim. It relies on the toxic compounds killing the rapidly dividing tumor cells slightly faster than they kill the patient's healthy organs. Biomedical engineers are approaching the problem from a different angle. They are replacing systemic chemical floods with precision-engineered strikes executed at the sub-nanometer scale. The foundation of this new architecture is elemental gold. In its bulk form, gold is chemically inert and completely ignored by the human immune system. To weaponize this sable material, scientists lower the gold into a nuclear reactor pool. Intense neutron irradiation transmutes the metal into gold-198, a potent radioactive isotope. By wrapping this induced radioactivity inside a biologically invisible shell, engineers can attack solid tumors from 5 independent angles at the exact same time. The objective is to eradicate the tumor from the inside out, without spilling a single drop of toxicity into the surrounding healthy tissue. Gold excels as a nanomedicine chassis because of its high standard electrode potential. It resists corrosion entirely, meaning it will not degrade and poison the host. Researchers secure targeting molecules to the gold using thiol bonding. This creates a direct semi-covalent link between the gold surface and the targeting protein, requiring roughly 190 kilojoules per mole to break apart. It eliminates the need for toxic chemical glues. But this engineered payload faces a strict physiological barrier. The human kidney can only filter metallic particles smaller than 6 nanometers, and these therapeutic particles are significantly larger. If doctors inject these larger particles directly into a vein, the body's reticuloendothelial system flags them as debris. The liver and spleen will permanently trap the circulating gold before it ever reaches the tumor. To bypass the liver and spleen entirely, oncologists use convection-enhanced delivery. They inject the radioactive nanodepots directly into the solid tumor matrix. They treat the bloodstream as a hazard to be avoided. Direct injection locks the radioactive payload exactly where it needs to stay. Once secured inside the tumor matrix, the nanoparticles use their thiol-bonded vectors to hunt down specific structural markers on the surface of the cancer cells. The targeting relies on precise molecular affinity. A cyclic RGD peptide will bind tightly to the Integrin receptors overexpressed by invasive glioblastomas, while the antibody Trastuzumab locks to bold onto HER2-positive breast carcinomas. Attached directly to the cell membrane, the gold-198 isotope unleashes the first 2 prongs of the attack. It fires a short-range beta particle that shreds the tumor's DNA, while simultaneously emitting a gamma ray that passes cleanly out of the body, allowing doctors to track the particle's exact position on a clinical scanner. The third prong utilizes the gold's optical properties. When exposed to a near-infrared laser, which passes harmlessly through human skin, the gold particle absorbs the light and converts it to intense radiant heat. This radio-photothermal flash cooks the specific cancer cells attached to the particle in a matter of seconds. For the fourth prong, engineers swap the solid gold core for super paramagnetic iron oxide, encasing it in a gold-198 shell. When an alternating magnetic field washes over the area, the iron oxide core rapidly heats up. This localized hyperthermia reoxygenates the dense tumor core and paralyzes the cell's ability to repair the DNA damage caused by the beta radiation. Finally, the extreme atomic density of the gold acts as a radiosensitizer. If the patient receives standard external X-ray therapy, the gold core amplifies those incoming X-rays, triggering a lethal cascade of secondary electrons right at the cell surface. Between the targeted beta radiation, thermal destruction, paralyzed repair mechanisms, and amplified X-rays, the targeted cells face complete structural collapse. This mechanism has already moved beyond theoretical physics and into phase 1 clinical trials in veterinary medicine. Researchers tested these nanodepots on companion dogs suffering from invasive, spontaneous prostate cancer. Visualizing 24-hour biodistribution, over 80% of the radioactive payload remained strictly localized inside the prostate tumor, with near-zero accumulation in the liver, spleen, or bloodstream. Over a 28-day timeline, tumor volume sharply plunged, achieving massive reduction with 0 systemic toxicity. Driven by these results in large mammals, research institutions are accelerating their investigational new drug filings for human clinical trials. These veterinary outcomes demonstrate that engineering therapies at the atomic level can create site-specific nanodepots that control solid tumors without triggering systemic toxicity. Beyond the surgical suite, this same radioactive isotope tracks fluid dynamics through the heavy steel pipes of massive petrochemical refineries. We will explore how nanophysics is optimizing heavy industry and deep-sea oceanography in Episode 4. Subscribe and stay tuned.